This is our seventh seminar in this fall 2019. Today we have speaker Dr. Ming Gao. He's talking about sweet potato breeding in genomics. Before his presentation, I would like to give his brief introduction. Dr. Ming Gao, he's senior research scientist at CARC, and he received his PhD from Penn State University in plant physiology. He did postdoc research at Iowa State University and Plant Biotechnology Institute of NRC Canada, and worked as a visiting scientist at laboratories of CNRC France and National Biological Center Germany. He is the author of numerous scientific articles in internationally known peer-reviewed journals, including a featured article in the high-impact journal Plant Cell. He also published book chapters, and he has also patents. He's also the recipient of more than $10 million of research grants as a PI or co-PI during his research career, including about 1.5 million at PBAMU. He conducts research in the areas of starch biosynthesis, storage route formation, breeding, and genomics in sweet potato. Let's welcome Dr. Gao.
Thank you. I guess we don't have many people interested in sweet potato, but anyway, we'll just make it informal. We're talking about. It's not wood. Let me see. It is on. I think, well, I'm here known as cucumber and the sweet potato guy. So I think my colleague have been tired of eating cucumbers. So this would be the last thing I'm doing the cucumber grafting production. So next year we wouldn't have any, but there will be some sweet potato coming. I've been working with the sweet potato for the past 10 years, but although I did not eat too much sweet potato, we're talking about more about my breeding and also the genomics work, mainly here. I have a lot of high-resolution pictures, so it gets to the computer very slow. Sweet potato, it is economically very important crop. It is the 7th largest crop in the world by production now. And it can be used where mostly we're eating as the storage roots, but some of those varieties we can use it as a vegetable. And it's very popular among the Asian populations here in Houston. Now, of course, we can see ornamental sweet potato in almost each and every subdivision in Houston. But also, right now, half of the sweet potato produced worldwide are used as a feed. We actually have another institution in the AM system, the Texas AM at Kingsville. There's a group developing sweet potato for feeding for rabbits, the meat rabbits. That's the two groups working in the Texas AM system. This is a map of world production. 75% of the world sweet potato are produced in China. The rest of those, about 25%. China used to produce 85% of the sweet potato, but now it drops quickly. United States takes about 5 to 7%, but it increases for the past 10 years by 10% every year. You see the dots there. So in many of these African countries, sweet potato is still used as a staple food. And currently, McKnight and the Gates Foundation are promoting the orange-fleshed sweet potato to replace white-fleshed sweet potato in African countries. And actually, it did not go well. as you know, the main reason is that our stomach is the most stubborn organ, so that it's very difficult to change it. Oh, it goes. This is the production from in the States. As you see, North Carolina is the #1, followed by Louisiana, California, Louisiana, and Mississippi. Texas, every year, that's about 15 tons. It's a small number, but there's one part that's more than that, produced by the prison system, which is not counted. So it's mostly in one county. The second one, that I was told that the prison system actually produced more than that county, but it's not included in USDA's statistics. Although it's a small, I'm going to say, although it's a small, but it fits our rule. If had it been similar to Irish potato, we wouldn't be in a position to compete with College Station. And right now, we have 2 small universities here in the system. I just mentioned the Texas A&M Kingsville. We're working with sweet potato. It is also biologically very unique crop. It is a hexaploid. It is one of I think it's the only hexaploid that producing storage roots. The potato is a stem, storage stem. So this is a root. So that we are biologically very significant and unique, but we do not know much about its genetic control of the formation of storage roots. This is a working model I summarized based on the literature. That's for my work, for my research work. So from this here, you don't need to know the details, but you know that we know very much about the physiology, but we do not know much about its genetics, particularly the control of storage root formation. Oftentimes, farmers would observe that you could see very good growth of vines, but you dig it, you don't see any storage roots. And if you don't do it well, there is not much harvest you can get. That's because the formation of storage roots, it's not genetically determined. It doesn't mean that you put the vines there or slips in the ground that you can get sweet potato. Sometimes you can get nothing. I'll show a picture where we'll compare. In some cases, if it's not done right, you don't get much of a harvest. This is the newly developed technology. A group of farmers in China started and invented a new technology. They grow sweet potato as grapes. So that these sweet potatoes, instead of growing in the soil, they're hanging on the shelf. So that those metal bars there, that's all the vines on top. So these here, this one has an advantage. You can harvest 4 times a year, and this can be continuous, and avoid a lot of soil insect problem. But also that you can expand to the cold area. But I set up some experiment trying to replicate it. In the back of a car, I failed miserably. I don't get any roots there. So I have to learn how they did it. Well, this is the left. That's the popular varieties we grow in US. And you see the three major types. The white flashed top, the Buga, all we got, almost 90% we buy from a grocery store, a Buga. And then the purple one. So it has an interesting feature here. Now, from the mouthfeel, the purple one's the worst. It's very dry. If it's rough, it's not good for baking. The white fleshed has the best mouthfeel. It can be well, in plain English, you can make a mashed potato very well. That's the reason it's so difficult for McKnight and Kids Foundation to promote orange fleshed sweet potatoes in African countries. So nutrition-wise, it's a reverse. The purple sweet potato has the best nutrition value. So what's good to your mouth may not be as good to your health. Now, I'm going to give a little bit introduction on the purple flesh. So it contains anthocyanins. Purple sweet potato has been around since, I guess, the people started sweet potato as a crop. It has never become popular in the world, and it was not promoted by growers. So recent years, the purple sweet potato is promoted by nutritionists, by medical doctors, because of these anthocyanins. The anthocyanin content is not as much larger than other fruits or crops. See, the purple corn has the highest, but the anthocyanins in the sweet potato are acylated anthocyanins. So it has a special effect on colorectal cancer and has risk reduction for cardiovascular diseases, particularly for the so-called metabolic syndrome. That's the one. So that it is now promoted by nutritionists and by medical doctors. That's why it's gradually become popular. The anthocyanins here has different colors. The color can change between among the 6 groups of chemicals, depending on pH. I guess this is a challenge to Dr. Myers' group. That's the reason why people are developing different types of products as a process, because the fresh type is still so difficult to make it popular. But this is a process, particularly in the noodles. They're very popular in the Asian countries right now, and the cakes, and then the bread. This one here, you can buy in Houston. It's imported from Sri Lanka and the Philippines. Also these ice creams. So now, because of its popularity and medical use, USDA has a special rule for organic. Anything, these juice used as a food color, no matter how purple sweet potatoes are produced, it can be labeled as organic, as long as you use the juice. It tells you how the importance, medical importance of this anthocyanin from sweet potato. Now, we got a problem. That's the reason why we're doing breeding. So first, the texture. It's very dry. You can see from this picture, the section. It's very dry. When baked, it's more or less like you're eating rock. It's very difficult to make it as a fresh type. The other thing here in the southern Texas, weevil infection. This is the reason I usually don't bring much of sweet potatoes here for our colleagues to share. So this is we're in a very heavy weevil infestation area. It's quarantined, so that means I cannot bring any sweet potato north. So this is also the initial breeding goal. So let's see if we can improve the texture and also weevil resistance. think I lost one picture. Okay. Well, this is the picture here is still in our field right now. I'm using this picture to introduce how we do the breeding. So sweet potato is hexaploid. So it's not a complete allo hexaploid, so that it displays very complex hexonomic inheritance. This is, well, it's more technical, but it's always just to say it's very complex inheritance pattern. So what we do here, and it's also an outbred species, which means that you have anther and a stigma here, but the pollen cannot self-pollinate the stigma here. So what you get here, it's called self-incompatible. Also frequently cross-incompatible. Sometimes you see this variety could not be crossed to this variety. So what we do here, that when we get the seeds here, we know that it is the mother, but we do not know which one is the father. And then these one seeds, each seed will be a hybrid. Once you grow it, and if you lose it, there's no such line in the world anymore. Because all the sweet potatoes are vegetatively propagated, so we call it clone instead. In sometimes we do not call it variety, we call it clone. So this one, we're doing a back cross. These because these are the selected lines. We're trying to cross back to the this resisto, which is the so far from my evaluation, it's the best weevil resistance. But you still, in our climate, in our soil, we can only get about 60% of intact without a weevil infestation. That tells you how heavy the infestation in this area. Now, how we do the selection. So this is when we grow it. So because we're looking at the texture and weevil infestation, this is how I do the selection. That's it. Literally, you just stay in the field. Take every line, this is a hybrid, you cut it, then you can feel the texture, whether it's tender or heavy. That's very easy to tell. And then you can see it, whether you get to see this one, it's such a heavy infestation. And we just destroy this line. So this is how we do the selection. So these here, these are the two lines I selected that's in 2016. This one is a weird line. You see, it's a white flashed background, but you do see the purple. But this one, when we do the evaluation this year, this purple color disappeared. So this one, I'm going to eliminate this year. So this is the process of phenotype selection. It's a very laborious, inefficient process. So this is the one we bring in genomics and also new technology in, trying to make the breeding process more efficient, so-called marker-assisted selection. It's called the marker-assisted breeding, MAB. So this here, this is illustrating agarose gel. So you see these two bands. It's bands here. You see particularly the small bands. This serves as a marker, so that this band is always linked to a resistant trait, so that when you select, you can grow at the seedling stage. You take a piece of leaf, you just extracting it, and then run the gel, and then you're going to see if this is linked. So this is a heterozygote stage. Then you can eliminate a whole lot. For this selection, each year, I can only handle about 150 plants. That's it. Right now, I still have 600 seeds. That's literally 600 hybrids that waiting for selection. I cannot do it. Each year, I can only do about 100 to 150. But this one here, when you run it, because most of those, previously we run in the gel, agarose gel. Right now we don't. We automate it. We can put it on the chip. Okay, we'll show it later. So you can screen 2,000 individuals within a month before you get these seedlings to the field. So it's a very efficient process. Right now, it has a wide applications in breedings of the major crops: corn, maize, rice, wheat. But in sweet potato, we're still at the stage of development of markers. This is slow. Okay, this just to show the timeline. I started this breeding in 2013. I evaluated a whole bunch of germplasm, and we found 3 lines that has a relatively better resistance against the weevil. But better, it means that you only have about 60% of intact sweet potatoes when we grow. Some of those lines that you grow that essentially 100% are infected, and essentially you have to throw it away. So that's the major reason. Every year you see, you saw me growing sweet potatoes, but I rarely brought any sweet potatoes for you to taste. That's one of the reasons. A lot of lost in the field because during the breeding process. Now this year, because we're doing the selection, so we do not use any chemical treatment previous years. So this year we do have some good harvest weevil-free because we're using chemicals. Now, I'm sorry. Oh, this is difficult now. Going back. It does not want to go back. Okay. This is the chemical that's a soil treatment before we making the bed, and then this one we do every 15 days for 6 times. Even under this treatment, for some lines, it does not guarantee 100% weevil-free. So now this one here, you're going to have a question. What about organics? So I think last year I said it here. I'm going to just basically summarize why we use it. So I would like you to hear a different voice about organic. So organic, the name organic is not a scientific concept. It came out of social movement. And actually it kidnapped USDA to come up with a set of regulation and so-called certification board. The certification board is not like FDA. It consisted of about a dozen stakeholders, only 1 scientist, so that all those labels that you put there are decided by one scientist. So it's essentially voiceless. So it does not mean better. It does not mean safer. And it only means that the label only means that it was produced without using certain synthetic chemicals. That's all. It's even not, it does not equals to pesticide-free or herbicide-free. So that has to be distinct. Okay, and also I would say here, if you visit some so-called organic farmers nearby, I would guarantee you never going to buy organic labels, okay? I would tell you that. It's getting to see that. So right now, the concept of organic, I would say it has gotten as crazy as our compliance on office here. But that's the side. We can debate on that side. So this one here, we see also the weed control. We have very bad wild amaranth on our farm. And this is passive. USDA putting a lot of money every year trying to combat the wild amaranth. So that we actually this year we got the success by using this lay-by and post-emergence spray. So that is now. This one I can handle by myself over weeding. Other than that, you see we usually after each rain, 2 days, all your plants will be buried in the weeds in the summer. So this year you see it's clean. So that's because thanks to this herbicide. Without this, the sweet potato will usually buried in the weeds. Okay. Now we set it up. You see here where evaluate this serves as an initial evaluation of the yield that we're going to buy 3 groups. Now I will put it there. So this is the selection here. I see. P that stands for purple, actually, because our university's abbreviation is PV. So you add V there, that becomes PV. This is all those years that in 2016 I selected 3. Last year I selected 1. That's say 18. So 19 stands that's by custom for sweet potato breeders. We use as a C because this year we're testing the resistor as a reference here. So this is the resistor here. As you can see, this is PV20. So when you take a look at the volume growth, so this is the PV1 as a spread. But this one here, number 2 here, it's bunch to spread. But for sweet potato, you don't have to worry about viral infection. Under the Texas climate, you have 1 week of high heat, dry heat. It's going to be cured. So the sweet potato has a self-curing ability to get rid of those viruses. But this one does not really have much of infection. This is a new line that grow at the beginning as a bunch, but it's not. It has some light susceptibility to viral infection. Now the problem is the leaf eater, those grasshoppers that make a big problem. So this one usually the favorites of grasshoppers. So and then the next one is this one. Whoops, it's okay. This is actually what we have. This is a resistor. This is a commercial line. So we put it here. You see, we use the rat damage. We have a lot of rats in the field. Okay, now the rats loves this. Now loves this one most. So it tells you. We just got a question before the seminar. This has a lot of free sugars before curing. So, and then this one, I'm sorry, I have to go back once. It's coming. This one, almost free. Rats did not even touch it. Although these lines are grow side by side with this one, the rest did not touch it. This just goes crazy. I should have a lower resolution. Yes, because of a free sugar. Now, sweet potato, for some sweet potatoes, that you don't get this sweet taste until they are getting cured. So it has a thermostable amylase. When you during the curing process, when the temperature goes to about 70 to 90 degrees in a range, that the thermostable amylase will cut starch into sugars. That's why you get the sweetness. So the longer you bake or you cook, the sweeter it gets. So these one you can see here. So these two are the favorites of rats. It tells you they have a lot of free sugars before the curing. This one in the middle, it's not much damage. Now you also see that the new lines have a whole bunch of 6 to 20, sometimes, but the size of a very small. And these here, the one you see here, the sweet potato numbers and yield are very close to the reference, to the mother. But this one here is much less. The fruit number is smaller, and also the yield is less. So that because of and also even under the chemical treatment, pre-cultivation treatment and during the growth period treatment, we still have 5% of infestation. So these one here, we're going to probably use it as a germplasm. We'll no longer go any further. So we're going to work on these two. These one, because initially it's under the background of the white flash. It has some anthocyanin synthesis, but now it's gone. So this one we're going to eliminate. On this side, I put it here. There's a reason right now we have the YouTube, the social media. Somehow it's a very strong impact on farmers and also garden growers. A lot of people, I guess for 30 seconds of fame, they put a lot of videos on the YouTube promoting growing sweet potato using plastic mulch. I had some doubt, but I saw that this year we included one line, one rule to see what's the effect. It turned out it's very detrimental. When we harvest, when you put all the grow side by side, every treatment are the same. The plastic mulch essentially prevented storage roots formation. You get very little harvest. See, this one, we got about 3 or 4. For some of those, you pull out nothing. On the other hand, the vines grow so beautifully. So this one here, so when we try to use this mulch, try to solve a problem of weeds, you create an even bigger problem production-wise. And then this is not scientific. And then you see YouTube has a very big impact on growers. This is wrong. You cannot use mulch for sweet potato production. Don't ask me why yet. I just tried it the first time. Come on. I skipped one. Please. Okay, this is perhaps summarize all I got for the, well, a lot of work for the past 7 years. So this is the mother. This is, this is one of the parents. This is the resisto. This is the purple sweet potato lines we use as a mother. That's from Burma. Now, this is the selected. When you see here, this is original that we produced every year. Now, the anthocyanin content is the highest among all here, but the texture is pretty bad. Okay, now this is the one that we selected, and you see here the color much less. So it shows you a reduction of anthocyanin synthesis. Now this one Well, a little bit, but there's probably the photographic effects. They are the same, but the size is smaller. You see, this one here has the commercial quality. The skin is very smooth, and then the size and the shape are very desirable. Now, this one here, this is high sugar, high free sugar one. So it's very light. So and also it's very sweet, and then it's the least weevil resistant. Now this one here used to have during the first 1 or 2 generation, we would see some purple color. Now during the field evaluation, now here it's gone. But the texture is very good. It can be comparable to Irish potatoes. So you can see here the anthocyanin goes down. The texture goes up, for more desirable. So for whatever reason, genetically, the anthocyanin synthesis in sweet potato storage roots is tightly linked to the texture, or mostly structure and dry matter. So there may not be a way to break it, to come up with a sweet potato has a higher anthocyanin content, but also favorable texture. So these might be as good as I can get. Have a little bit lower of anthocyanin content, a little bit improved texture. And we don't want to get to this stage. These are all from these 2 parents. But this one we collected, but would serve as kind of a genetic material for me to do some kind of a genetic study to figure out what's the loci that impact the anthocyanin synthesis. So this is the one we're going to go further evaluation, and these probably, but the other 2 will be eliminated. As I just mentioned, I still have 500 hybrid lines waiting there for me to evaluate. So how we come, why the other thing, I'm doing the breeding, why I'm doing another one, so-called the genomics. This is how, I hope I can use these slides to explain what heck is genomics and marker-assisted breeding. Now, this is a sweet potato genome. It's an illustration. Now we human beings are diploid. We get one set of chromosomes from a father, the other set get from a mother. On the other hand, the sweet potato, like you have 3 fraternal triplets stuck in together. So this is one set. This is another set, subgenome. This is another one. They have 3 sets of these chromosomes. So one of the chromosomes, so you got here, these 2 chromosomes called homologous pair. That in human being, one from father, the other from mother. So on these here, you have differences here that we call it allele. There's a genetic variation, we call it allele. So if we're looking for is allele, the difference between these 2 that linked to QTL, quantitative trait loci or locus. That's what we're looking for. If we can find this, then we can just do the marker-assisted breeding. Now, how we do it? Do the screening instead of growing in a farm and then you take them out and cut it. We can do a PCR amplification. You get everything together and then do a next generation sequencing so that you can get to know which line has the trait. So link the molecular marker with the phenotype. That comes to the genomics. So this slide summarizes all we have done since this project got funded by the CBG. So the green part has already been completed. This one here that depends upon funding, we're going to continue. So this is the first step. We sequenced 3 species, the messenger RNA. That's everything we're trying to figure out what's the gene there. So these one sequenced at Indiana University. So and this part we sequenced using the amplicon. That's a PCR product. So we sequenced here in my lab. So this is the sequencer, next generation sequencer. This is it is a robot that we prepare the library. This is the lab chip, so that we no longer run in a gel. So we put it there, we can get the gel effect, everything. So we're trying to automate. So here we're going to add another piece of a robot so that later we no longer need to prepare it by hand. Everything can be prepared by a robot here. So right now, this is the marker primer pair we got. It's about this number that covers these loci. That essentially cover most of the differentiated genomes. This we don't need to know that this is very tedious, strenuous work. So these varieties, this is diploid relative to the sweet potato. Right? Currently, I'm the only one in the world to have this line. I initially got it from USD germplasm site. They lost it, so I have it. And I got another one from Florida. So the 2 accessions that in the world, I'm the only one to have it. And we also have the only one to have the sequence here. We're going to continue to work. Now, these data are publicly accessible. This is used to be hosted on the supercomputer mission in Indiana, the National Center for Genomics. Okay, now this is our website. So this one, they got this website up and running in 2 days. And until last week, because we have to pay for it. So because the money runs out from the grant, now it's no longer ineffective. This one here, this one still, we are still working it. It's down by people. And when he left, so we don't have anybody. It's already 2 years. That's the difference. Hopefully we can get help to get this website running, because this is perhaps the best publicity, because these two databases, we are the only one to have it. Close. So this is the one that we're illustrating how we get this, how we do the genotyping. So these, illustrate 3 chromosomes. We'll see here that's from those fraternal triplets. So with here, this is here indicates this kind of a family. We have different families, and then this is a family label. This is the true allele label. Now, we're looking at the ratio here, the genetic segregation ratio. This is what we're looking for when we do the doing the genotyping. Now, this one can be, what we do is we amplify a whole bunch, and then we can, right now, we can, for each sequencing run, we can get 1 million different reads, more than that. Then we can assemble them. We can, the program can automatically, you set up the reference, so you get this, it can automatically tells you what it is, the ratio. So now let me quickly go here. This is a different situation. This is what we're going to get. After the run, after the computer, even the computer is going to run all those 1 million reads. Takes about 12 to 20 hours to run, and then you got reports like this. So this is true. We tested some of those markers. So it tells you here. This is essentially it's a heterozygous. Now the reference is G. The residue as G, it's changed to T. This is a variant. Now, this one here changes more. Now, we're looking for these type of markers that linked to the anthocyanin synthesis. Then we can use these as a marker to tell whether the segregating populations, which one we would have this marker, then it must have that phenotype. That's what we're doing. So we're essentially towards that way, but probably about 60% are done. It depends. I think I did not get the funding for this year, so we will have to find other ways to get grant funding. We'll see. Because we have all these data sets, so we kind of sidetracked. We did a phylogenetic study. So these are all the variation indicators. This is a very highly large, big piece of DNA. We put it together. So don't worry about this technicality. So we see we just conducted a large scale, pretty long, tedious phylogenetic studies. So we're trying to figure out where possibly sweet potato was originated. So let's get out here. So after this analysis, this is a summary of those analyses. Let's see here. Afterwards, we get a gene tree that tells, this tree tells you how these different genes are where originated in some sort of references. This is a bunch of genes from Morning Glory. This is the Ipomoea Batatas. This is Tennessema. This is Trophida, one of the progenitors. So now we got another type. We'll just quickly. This is another gene tree. So what are these used for? Allow us to figure out a species tree. This tree tells you how sweet potato here were possibly originated. This is how, so I'm going to see how this tree fits with the species tree, how fits. You can see two sets of lineage. Those lines represent a lineage, a series of ancestors. So how it fits those two trees. Matching on the side. I should have made these lower resolution. Now, what it says here is this hexaploid genome, sweet potato. Okay. Here, the sweet potato was possibly derived from a hybridization of a tetraploid here with a diploid, Trophida. And then this tetraploid ancestor were in turn derived from hybridization of two additional tetraploid. They are all derived from one species. So that the three genomes, or so-called fraternal triplets, were derived from close relatives. And then that's why they are so similar. Those three genomes are so similar. So these are called the species tree, explained based on molecular data how they the sweet potato is derived. So technically, it's very complicated. Actually, these were in a revision for submission. But the first time when we submitted some of those data, actually, I misinterpreted it because by training, I'm not a phylogeneticist because we have the data. We just venture out to do the phylogenetic study. So that's the end. I have to acknowledge NIFA for the CBG grant and also USDA Stoneville. They supported partially for the sequencing of the sweet potato. And then my collaborator, Dr. Ketan Kaitis at that time at Indiana University, and then Dr. Fonte Sariano and Dr. Michael Kabrum, and these are the people worked on there. But here, I particularly, I would like to give a special thanks to this student, Gerald Thompson, and also our Willie DeWall. Without them, every year the field work, personally, I cannot handle it. Now, this kid got a very good job in Honeywell right now. I lost him, so we're trying to recruit a new student. I just usually I put out ads for my position. This year I only got one student from our college, and I mistakenly thought it is a boy, would be a boy, Morgan Green. So when he interviewed, came to my office, so I have to laugh at it. I made it wrong. I did not know him here. Otherwise, I would have recruited you. Because I usually don't get any applicants from our college, as long as I put the name there that we have to work in the field. So, any questions? I think I made it on time, right? Okay, sure.
I know one of the problems with data globally is getting them to other locations in a timely manner. I know that whole shiploads can be lost because of delays in that. Is there any relationship with the texture and the longevity of the sweet potato? In other words, do the ones that are more fibrous, do they last longer than the ones that have the better texture?
No. The longevity is mainly associated with the free sugar. The higher free sugar it contains initially without curing, the more difficult to preserve.
In the, I'm sorry, is the sugar lower in the more fibrous, less textured?
No, not necessarily.
Okay.
Not necessarily. It is a separate trait. You can have say for purple sweet potato, it has a very tight, rough structure and high carbohydrate content. On the other hand, there are varieties which has very high free sugar. Some other varieties don't have it. So it's an independent trait, whether you have it or not. And actually, I just learned in the field, rats are the best to tell. They don't touch those without sweetness. So it's an independent trait. So right now, for all those sweet potato we buy from grocery stores are already cured. So that they're reasonable. You can, even at room temperature, you shield it from lights. Those cured sweet potatoes can be kept for at least 1 year. But it cannot be as long as those dried greens. That's the problem. So that's the reason why North Carolina right now grows sweet potato by 10% increase every year. The USDA ARS at Chapel Hill invented novel processing procedure so that they make fresh sweet potato into puree, and then they put it into the big gas can. That can be stored for 5 to 6 years. For most. So that's resulting in the big increase of the sweet potato cultivation. And those purees can be used in a lot of the soap thickenings and also ice creams. If you look at the back of a lot of ice cream, you'll see sweet potato puree.
I have a quick question. I'm sorry.
Go ahead, Dr. Kaz.
So is the anthocyanide concentration or the biosynthetic pathway, is it related in any way to the thickness of the skin?
Your question is the anthocyanin synthesis pathway
Or the
has something
biosynthesis-
to do with the thickening of the skin.
The skin on the
No.
no.
The skin thickness is independent, but where the distribution of anthocyanin has something to do with the pathway. So also, as you know, the anthocyanin is a type of phenolic compound. In some cases, the phenolic compounds serves as a pheromone for soil insects that make things even worse. We know all the enzymes and all the genes responsible for anthocyanin synthesis, but we do not know how the pathway is controlled right now. Did I answer your question? Thank you.
Okay, thank you, Dr. Gao. No wonder seeing you every time I went to the field, babying those sweet potato for a long time. You just mentioned one slide at the beginning is about the Chinese technique on growing those sweet potatoes out of, they don't need earth factor, because it seems that the earth factor is interfering what we are looking for here. The two things, the tender line and the texture, is the infestation by those insects. So I know you tried, but why not in the future thinking of trying that variety of, or the way that they do it, because it will make focus more on these two main factors, the texture and the
Okay.
yeah.
If I understood correctly, your question is why not try those type of grip type of cultivation of sweet potato here?
Yeah.
So the answer is I really wanted to try, but I failed miserably this year. So if we can do it, it can make it really a good exhibition. So you grow sweet potato like you grow grapes, so that you can see the sweet potato hanging down and bulking every day. And then underneath, you can have them over your dining table or somewhere in the field. That's really a good exhibition. But the answer right now here, I cannot because I don't know how to do it yet.
Yeah,
I will try.
That's also will match the new work with the new technologies and
Yes,
different
and
issues.
also very good for me to harvest materials to study how the roots are formed, why certain fibrous roots bulks.
Exactly.
We do not know. And it's difficult to harvest from the soil because the roots are going to be destroyed. So you're not going to be able to sample. But these can give me the chance to sample very precisely. But I don't know how to do it. Actually, we tried it before, but all failed. So right now, it's a trade secret. So that how they do it, I don't know. So I'll have to try to see if I can replicate it. Well, I will try. But this year, in the back of a car, I failed. So I'm going to put them away.
Yes, I have a question. The Inter-American Institute for the Cooperation on Agriculture partnered with LSU and North Carolina State University, along with the Ministry of Agriculture in Jamaica, their extension arm, in producing sweet potato because of the growing demand for the orange flesh sweet potato in Europe and Canada. Are you familiar with this partnership, and do you see opportunities for such collaboration here at Prairie View A&M University?
Yes. The I'm familiar and also have, worked with the main researchers, the Lepanti, Clark, Dr. Lepanti and Dr. Clark at LSU, and also groups in North Carolina State. But I don't see any chance for them to work with us. We're too small. And they wouldn't give us the chance. I would say it. I applied for, I submitted a grant proposal to McKnight, both McKnight and the Bill Gates Foundation, trying to get some funding. Where I did not get it. So it's a good idea to try to work with them. When you do the collaboration with this kind of skill, unless you have something to offer, people will be very polite. At this moment, we don't have concrete things to offer. So they might be polite to you, but I don't see any chance to work with them, frankly. In most collaborations, when if you are trying to make a collaboration work, you have to be in the position to offer something important to the collaborator. Otherwise, that collaboration will not succeed.
Here in Texas could benefit from producing the orange flesh, sweet potato, based on the demand in Europe and Canada. Do you see how there are potential benefits and how we can maybe educate our producers here?
No, I don't see any benefits because the local farmers right now facing the problem is not whether they can sell it or not. The problem here is that they cannot have their produce processed. We don't have processing facilities in Texas. If they produce too much, they're going to be in big trouble because the sweet potato can be rotten with over 24 hours if you don't process them. That's one thing. Secondly, we here, when you cannot ship or to some places else from Texas, all the processing facilities are in the Northeast, in the Virginia, West Virginia, and North Carolina. You cannot ship all those sweet potatoes to those facilities. So that's not any benefits, although you have the demand in European countries or Canada. Actually, Canada, they started growing sweet potato in Nova Scotia on the island. They succeeded in the cold weather. But here, that's not the issue. They are not. I don't think you can. They would like to give you the chance to have the collaboration. That's the first. Secondly, there's not much benefits to our local farmers. That's my personal opinion. Dr. Myers.
Just quickly, I guess as a comment more than a question, is that we're working with Dr. Gao. There's another part to this. This is the value-added side. And so in our labs, we're doing some things with Dr. Gao with the purple sweet potato, looking at starch. We're looking at sweet potato chips. We're looking at flour and using it as an ingredient and so forth. So as he's continuing his work, we're also looking at the value-added side of it. So we're doing that together, and hopefully we can come up with some ideas. And Dr. Henry, what you're talking about is maybe we can come up with some value-added uses that the farmers here can use it that maybe won't be very similar to what's already being done. So hopefully we can make that happen. So I just wanted to make that comment.
Any more questions? I think we can conclude here. Next week we have a seminar from students researcher. Dr. Carson will coordinate next week's seminar. Let's give a big round of applause for.
Thank you all for coming.